Pili are short, hair-like structures on the cell surface of prokaryotic cells. They can have a role in movement, but are often involved in adherence to surface. This is what facilitates infection and is a key characteristic of how bacteria can cause sicknesses, illnesses, or even disease. They are utilized by both bacterial cells and even viral cells. Conjugation is the process by which one bacterium transfers genetic material to another through direct contact. During conjugation, one bacterium serves as the donor of the genetic material, and the other serves as the recipient. The donor bacterium carries a DNA sequence called the fertility factor. Bacteria uses structures called pili to adhere themselves to other bacteria, making pilis important in the DNA transfer between cells. Since pili was described in the last slide, it is very important to bacteria again for this conjugation and DNA transfer. Without them, it would not be able to exist. So, an example of this, the ability of the S plasmid of Escherichia coli, also known as E. coli, to efficiently transfer DNA from an F-containing donor to an F-deficient recipient was the first described example of bacterial conjugation. And E. coli is known to cause a lot of food sicknesses that can cause vomiting, chills, illness, a lot of different symptoms for many people. E. coli bacteria normally live in the intestines of healthy people and animals. Most types of E. coli are harmless or cause relatively brief diarrhea, but a few strains can cause severe stomach cramps, bloody diarrhea, and vomiting.
Transduction is the process by which a virus transfers genetic material from one bacterium to another. Viruses called bacteriophages are able to infect bacterial cells and use them as hosts to make more viruses. In transformation, a bacterium takes in DNA from its environment, often DNA that's been shed by other bacteria. If the DNA is in the form of a circular DNA called a plasmid, it can be copied in the receiving cell and passed on to its descendants. An example of this is if a harmless bacteria takes up a toxin gene from a pathogenic bacteria, it too can become pathogenic if it incorporates the new DNA into its own chromosome. Another example of transformation is that it's used also to make large amounts of specific human proteins, such as insulin, to treat type 1 diabetes through artificial transformation. In transformation, a bacterium takes in The key takeaways from this video are that transformation is when a bacterium takes up a piece of DNA floating in its environment. Transduction is when DNA is accidentally moved from one bacterium to another by a virus, and conjugation is when DNA is transferred between bacteria through a tube between cells.